alterations in brain cholesterol homeostasis is increasingly being recognized as a potential contributor to neuronal excitotoxicity, a phenomenon characterized by excessive exposure to the neurotransmitter glutamate or the overstimulation of its NMDA receptors. This, in turn, can lead to excitotoxicity causing neuronal injury or death. Excitotoxicity is the hallmark of various neurological conditions, including Alzheimer's disease, Huntington disease, and status epilepticus. Seizures cause excessive glutamate release, which in turn stimulates NMDA receptors, triggering substantial influxes of calcium ions into the cell. This cascade of events, ultimately, results in widespread neuronal death, partially attributed to the presence of inflammatory proteins. Following an initial seizure, a latent period may extend over several years, during which synaptic plasticity and the reorganization of neuronal networks undergo changes, potentially giving rise to cognitive alterations. The reorganization of these neuronal networks makes them more susceptible to excitatory signals, ultimately paving the way for the development of spontaneous and recurrent seizure episodes. Proposed temporal changes in free cholesterol and 24-OHC levels following status epilepticus can be categorized into three phases. In the initial hours and days post-SE onset, 24-OHC levels decrease, likely linked to transient CYP46A1 inhibition, while free cholesterol levels remain normal. Several days after status epilepticus initiation, free cholesterol levels rise due to the absence of a negative feedback mechanism initiated by 24-OHC on cholesterol synthesis enzymes, and 24-OHC levels return to normal. In cases of prolonged status epilepticus, patients experience neuronal death, leading to an increase in 24-OHC release. Studies have begun to examine the impact of blocking brain cholesterol catabolism as a therapeutic target for patients with developmental and epileptic encephalopathies, or DEEs. DEEs are characterized by severe seizures that are often drug-resistant. These patients also have encephalopathy, which can indicate significant developmental delays and loss of skills learned during development, such as talking or walking. Examples of DEEs include Dravet syndrome and Lennox-Gastaut syndrome. In a mouse model of Dravet syndrome, researchers demonstrated that inhibiting brain cholesterol catabolism via CYP46A1 signaling using soticlestat reduced susceptibility to seizures. Further, soticlestat treatment was shown to reduce the seizure burden and improve survival in Dravet syndrome mice. Inhibition of brain cholesterol catabolism is currently being investigated in clinical trials for DEEs. The Phase 2 clinical trial ELECTRA examined the use of soticlestat in patients with Dravet or Lennox-Gastaut syndrome aged 2 to 17 years old. The results revealed a statistically significant reduction in seizures from baseline compared with placebo in Dravet syndrome. However, patients with Lennox-Gastaut syndrome did not show statistically significant reductions. It is crucial to ascertain whether cholesterol accumulation, resulting from the inhibition of cholesterol catabolism via CYP46A1, leads to neuronal death in humans. This is of particular significance, considering that inhibiting this enzyme has been shown to induce epileptic behavior, seizures, and neuronal loss in mouse models. Additional research is still underway to determine the safety and efficacy of this therapy in humans.